Okay, hi guys. So in the last class, before going for a break, we did explain about the whole thing about the GM crops, right? So we explained about the GM crops. If you would remember, we said about the first GM crops, the second GM crops, and also the third. And we also went into reference with the RNAi interference. I hope all these points are crystal clear to you and you have no doubts about this. Am I right? Okay. So from here, we move on into the next criteria, that is how to create a pharmaceutical drug, and that too a drug which is artificially created. We would look into the reference of insulin. So I'm going to refer in here with insulin. I hope it's very familiar to you about insulin. Okay. So insulin, if you would remember, what are the points you remember about insulin? We know that it is taken from the pancreas. Okay, so I'm going to draw the pancreas in there. Let's go for the pancreas. This part of the pancreas, I would like to call it as a tail part. So this is the body part of the pancreas. And we say that this part is exocrine and this part is endocrine. And therefore, the pancreas is said to be heterocrine. It's called as heterocrine mainly because it has got two parts, exocrine part and the endocrine part. Now here we are referring to only to the endocrine part. So I'm picking up the endocrine alone. So let's look at the endocrine. In the endocrine, I will find that there are certain patches like this, or I would find that there is certain cells like this, islands like this, which I would like to call it as islets of Langerhans. Now, if I would like to zoom on this, let's zoom on that. I would find that there is another set of cells in there. Now, this is a blood vessel. Don't worry about this. This is your gamma cell. This is your alpha cell. This is your beta cell. Now, this gamma cell is the one that gives rise to somatostatin hormone. Clear? And this one gives rise to glucagon hormone. And this gives rise to insulin hormone. So our reference is with the beta cells. So from where did I get the beta cells? It's from the islets of Langerhans, which is located within the endocrine part of the pancreas. So the question is, when I understand what is insulin and from where I can get insulin, the next question is, what is insulin doing in there? Okay, let's look into this. The insulin is a structure which is comprising with 51 amino acids. It has an A chain and a B chain, which we normally call it as the alpha chain and the beta chain. You can call it either ways. It's okay. So this is the one we call as the insulin. Potentiality of this can convert glucose to glycogen, which means that we will have very less of glucose in here, which is this blood vessel. This is what? Blood vessel. So the blood will be containing less levels of glucose thanks to insulin, which means that this hormone is hypoglycemic. So when you use the word glycemic, it means that it is something connected with glucose. Hypo, low levels. So low levels of glucose in the blood vessel or in the blood is possible because of insulin. That's why it's called as hypoglycemic hormone. So we've got an idea about that. Now what if you don't have this insulin? Then we contract the disease which we call it as diabetes. So the disease that we get from this is what we call it as diabetes, and it is none other than diabetes mellitus. That's what we get from a deficiency of insulin. So a deficiency of insulin will lead to diabetes mellitus. So this is the issue which we are going to face in the current society, or we were facing in the previous society. So people started thinking about it, how to get rid of this diabetes. Is there any solution that I can find out for this? I'm not going to rub this off because I need this again. So I'm just going to take these things off. Okay. So initially, there was yeah, way back to me, my forefathers, they might have had an issue because they were finding it very hard to get the insulin and then find a cure for diabetes. So Banting and Best isolated insulin from dog in the year 1921, whereas the Nobel was given in 1923 to Banting and MacLeod for demonstrating that they can administer insulin as a cure for diabetes. So once they found out that insulin can be administered, then started the isolation of the insulin. We thought of giving it from, extracting it from dead pig, porcupines and all that stuff. And if you would see, pig insulin and ours is only different by one amino acid. That's what I've written down on the end here. Porcine insulin, which is pig insulin, except for the change of ALA B30 to threonine B30, alanine, that's alanine B30 to threonine B30 in human insulin. So alanine in the B chain, which is in the 30th position, that will be changed to threonine in humans. That's it. That's a change in there, but even though it's a small minor change, but it's a change there. So that's how we differ from the pig insulin, but that was the one which we used to give for our earlier patients. But then it was found out that they might have, contaminations in there. So we started not using it. And then we thought about, giving it through a medication way by creating an artificial insulin, and we called it as Humulin. So once we thought about making a Humulin in there, then came the idea as to from where can we do this, how do we do this, etc., etc., etc. So that calls for the next idea in there. What we did was, we knew that it is extracted from the beta cells, right? So from the beta cells, we took the gene for that. Now this was done by a company called as Eli Lilly. That's also an American company. Okay, so they were the first people to do that. So they took up the gene from the beta cells, which codes for insulin, and they incorporated this into the E. coli bacteria, and they were expecting insulin to prop up. But what they got was proinsulin. They didn't get insulin. This is what they were expecting, but instead they got proinsulin. The question is, why is it proinsulin? Because proinsulin is almost 86 amino acid long, and this one is only 51 amino acids. It's a huge difference. So my question is, why did we get the proinsulin? Why not the insulin? So we dodged into it only to find out that even though the beta cells is producing proinsulin, inside of them there is the presence of Golgi bodies. These Golgi bodies will take up this proinsulin, clip off the C peptide and make it into insulin, the proper insulin, with only the A chain and the B chain. So what is there in the proinsulin? An extra C chain is present, which is 33 amino acids. So the question is, oh my God. So I've got a proinsulin, which is 86 amino acids, and they have an extra C chain in there, and I cannot process this because I don't have a Golgi body. Who doesn't have a Golgi body? The E. coli. They doesn't have a Golgi body. So the question is, how do I do this? So initially, what these people did was, they took up the genes, like since this is the gene that codes totally for proinsulin, what they did was, they took up the gene that codes for A, they took up the genes that codes for B, and they gave it separately to a batch of E. coli. And then in here they produced A chains, in here they produced B chains, then they added disulfides to it, and hence insulin was formed. And this insulin was called as Humulin, the first artificial one which we created. Now the problem with this is, it's a very tedious step, not easy to understand, not easy to do, and it's very costly too. So what we did was that in 1990, the company shifted using the E. coli. Instead of that, they started using yeast. Yeast is a eukaryote for information. So we can use, this is also unicellular, which means that division is comparatively faster. So they used yeast. And as a result of which, they could just give this gene directly into the yeast, and it will have the Golgi body, so it can cut it off and can form the insulin in there. Now the question is, which are the areas that I should remember about understanding the insulin in there. Now, insulin, like I said, has got another speciality in here. If you would look into the chain, look at the chain difference. We have the structures to be learned. So this is our proinsulin. This proinsulin is an E in the reverted order, and you could say that this is the A chain, and this A chain, this A chain is with 21 amino acids. This is the B chain, and the B chain is with 30 amino acids. And here we have the C chain. The C chain is with 33 amino acids. So you might ask a question to me that, teacher, it amounts only to 84 amino acids, while you said that it is 86 amino acids. Yeah, of course, I said that it is 86 amino acids because the one that is used in here and the one that is used in here is for using as a fixature in there. So that's the reason why I say that it is 86, not 84. Even though in totality, you could say that it is only 84 in there. So is these many points crystal clear to you? Thank you. So from there, we move on to understanding the structure in here. Now, who gave this primary structure an idea? That was done by Sanger. So Frederick Sanger gave this structure, or actually he was the one who modeled the structure. The primary structure of insulin was given by Frederick Sanger. Now, the point in here is, when you take the normal insulin, which I said these two are the only one involved, we would like to know where are they placing their disulfide bondages? How are they linked? See, you can find the linking in here also, but I would like to show it in here because that structure is primary. This is the proper insulin. So once this comes in here, we can find that in the A chain 6th amino acid and A chain 11th amino acid is having an intra connection in there. See that? That's an intra one. Now here I have another connection which is between inter, and that is between the 7th amino acid of the A chain and the 7th amino acid of the B chain. Here we have it as the 20th amino acid of the A chain with the 19th amino acid of the B chain. So this is how you get the chains. So for that, I think I could just rub this off. We'll just clip this. Is it fine? So I would like to say how the chains are, because that's another question which we normally get, and that is A chain and the B chain. Let's pick it up in this manner. So it is in the A 7 to 7, they have a disulfide connection, and here we have it in the 20th and in the 19th we have another disulfide connection, and here in the 6th and the 11th they have an inter one. So this is how we get the connections of the disulfides in A chain and the B chain. So I hope this whole idea is clear, and this is how we got making into the humulin very clearly. So right now in 1990, like I referred, in 1990 we have made it through with the yeast, and this will be having Golgi body, so I don't have to worry about introducing it separately, etc, etc, etc. I can just give it directly. So I can just put the code from the beta cells, I can take it directly, and okay, I take it from here directly, and from here I would take this into the yeast directly, and finally, since there is Golgi bodies, I will definitely get my insulin in there. So I got my insulin. This is the insulin there. So I've got the insulin in there, and I would call those as humulin. Simple process now. Earlier it used to be very tedious, now it's very simple. So this is how we get the insulin in there, and that's how we have made our first drug in there. So with which we have come to the end of almost half or almost 3/4 of the chapter. Now we move on into understanding about gene therapy. Let's look for gene therapy. There's a famous question there. So in this question, we get the idea that is a first clinical gene therapy was given for treating. Now, I would like to place this kid with a name, and her name was Ashanti De Silva. She was a 4-year-old kid. And the successful thing happened in 1990. I don't know why 1990 is so much jinxed. So many things is happening in there. So in 1990, we've got a first patient cured from this so-called gene therapy, which was ADA therapy. Now you would look at it. Which is that disease? Adenosine deaminase deficiency was cleared in there for this little kid in there in the year 1990, and the kid's name is Ashanti De Silva, and she was only 4 years old. Now the question is, what is this whole menace about? So we look into that. Let's have a look. We need the basics of understanding. So whenever a cell has to replicate, see, this is one cell, it's ready to replicate into two. Okay. Once a cell is determining to replicate, it requires the replication of the DNA also, right? So if this is my DNA, I need 2 copies of DNA. Am I right? Then only I can have a 2-cell division. So I need the DNA also to be replicated. For that, once the DNA is supposed to replicate, I know that it forms a replication fork, right? We have learned in genetics, and we also have these people opened up in there. So we have to require the nucleotides from the pool. But the point is that in a normal cell, we do not find this is not present. Instead, we will only find the question is, why do I find only riboxynucleotides and not deoxy? Because that's how it is. Because there is an enzyme in here, which I would like to call it as RNA reductase. If this reductase is there, then only, see, ribonucleotide reductase, RNA reductase, if I have that only, then only can I get this converted to this format. Thereby, I can get this in plenty. That's how it is. But the problem is that if this enzyme is also already in there, teacher, why can't I get this? The problem is, again, this is locked by another person called as DATP. So this person says, don't act until I tell you. So now this is blocked. So this conversion is not possible. This is being blocked. So I don't have any deoxyribonucleotides. So even if I say that to divide, division is not possible. The question is how to remove this DATP. This DATP is removed by the entry of ADA. So once ADA comes into picture, it goes up, it takes this person out, and as a result of which, once DATP is removed, so I'm just removing it out. Let's look at it. I'm just removing. I think I'll just redraw it. That's better. Okay. So this is the enzyme. Let's see. So I have removed the DATP. So DATP is removed, which means that this enzyme is plenty right now. So I don't have this. Okay. This is also not there, which means the deoxy is there. So I can get this converted. See, this is how it normally goes. Now the problem in here is I'm having a deficit in this ADA. Oh my God. So ADA is deficit. So what happens is I will not be able to replicate. That is a basic understanding in there. If you know this understanding, and if this understanding is very clear inside of you, then we can explain the process. The process goes very simple. Like when ADA is not there, like I said, replication is not possible. This is happening inside the lymphoid stem cell. Now, what's the specialty of the lymphoid stem cell? This is the one that is responsible for the production of T lymphocytes, B lymphocytes, and NK cells, which we call it as natural killer cells. So the idea in here is I have the T lymphocytes, I have the B lymphocytes, I have the NK cells also in there. That is the one from which the prior one, this is being formed from lymphoid stem cells. Now the question here is, if the lymphoid stem cells cannot divide, forget it. I won't have this, I won't have this. Which means that there is no T lymphocytes, there is no B lymphocytes, which means that I'm having the disease which we call it as KIT, which is severe combined immunodeficiency. So if I have this, then the issue is that how to cure this. I just have to supply the ADA, right? So what I did is I took up the ADA gene. Okay, this was done in the year 1990, and we call it as gene therapy. Okay, we took it into a retrovirus, disarmed it, and then we supplied it into the bone marrow. successful only in the year 1990, and that was done only for that small kid in there. After that, even though we tried the same step, it never worked. It was like once in a blue moon it happened, something like that, or it was one time luck, or you could say that one chance. So it was just a chance in there that this thing happened in 1990 just for one kid. After that, this never happened. So what we did is we rerouted it. There is no other way to cure this. So we thought of introducing this into a lymphocyte. That was our next area of thought. Why not we introduce this into a lymphocyte? Now, if I introduce this gene, that is ADA gene, if I introduce this into a lymphocyte, then the issue is that this lymphocyte has only a certain age, lifespan. That might be 80 to 90 days of lifespan, the max. So this recombinant one, because this now becomes a recombinant lymphocyte, isn't it? Because it's not the normal gene composition in there. So I have a recombinant one in here. So once this is recombinant, that will be having only 80 to 90 days at the maximum, which means that I have to reinfuse it with intermittently. So after every 80 or so, per se 90 days, I will have to introduce the recombinant lymphocytes into the body stream to make sure that this patient doesn't suffer from SCID. Now the issue is that how is this lymphocyte helping out? The lymphocyte is the one that keeps on wandering. So this person will never ever remain in one position at a time. It's like a gypsy, keeps on moving from here to there, from there to here. It keeps on wandering. It loves to go to different places. So this person, even I'm very much interested in, being the gypsy. That gives me the idea of lymphocytes. So it will definitely move into the lymphoid stem cell. It will remain in there for some time. Like how you go for a gypsy, they go to the place, they stay there, they look in their environment, they study about the place, they learn a little bit of here and there, their culture. Then they say that, this is enough, I've got enough of it. So the same manner, this person will also go in there, stay for some days, look around, and he'll find that, okay, this is T cell formation in there, this is B cell formation in there, there is NK cell formation in there. He'll be looking at it from there. By the time or till the time he's in there, this gene is operating, which means that these will be formed, which means that this will be cured. That is how we are doing it in a temporary basis right now. We can't do it in a large scale right now. Is that point so far clear? So this is how you would cure with the ADA. So that is about ADA SCID. This is called as ADA SCID because it is due to, this SCID is definitely due to the deficiency of ADA, adenosine deaminase enzyme. So with which we end up with the ADA also. Now the last and the least area of this topic, we are winding it up. I know it's been tedious for you. I understand that perfectly. So from there, we'll move on into the only two area that is remaining, that is the transgenic animals. Let's look at the transgenic cow in there. So the transgenic cow, it was the cow. See, when you drink a glass of milk, okay, let's pick up. This is a glass of milk, cow milk. What do you get? We get proteins. That's what we say. We get proteins, yeah. So, but in human, let me put me in there. I don't get enough proteins. That's why my muscles is not that strong. And when you go for a gym, now I'm going for a gym. I'm hitting a gym. Okay, let's put the dumbbells in there. Or a weight. Okay. So I'm going for the gym. I'm going to hit the gym. So this is my gym in there. Okay. So when I go to the gym, the person says that, oh no, you don't have proteins. Your muscle is totally loose. As if I don't know. So I say, yeah, but I'm drinking milk, on a daily basis I'm drinking milk. I say, no. You have to go for something which we call it as powdered protein. I hope all the gym people will agree with me that the master suggested them or their teacher suggested them that they should take up powdered protein. Have you ever thought about what is powdered protein? That's just to boost up your system. Yeah. So how do I get this powdered protein if I'm drinking only a glass of milk? Very simple. You have to take the milk from it, make it an enriched milk. How do I make this milk enriched? You have to see that I don't require the protein that comes from the glass of milk of cow milk. Okay, let's pick it up. So I won't get the protein which is essential for me from the cow milk. So how do I make it enriched milk? What is the protein that I'm looking forward to? I'm looking forward to human alpha lactalbumin. Now this is the protein that I'm looking forward to. But unfortunately, my cow milk is having very meager amount of this, which means that there is no other way I can get this. So what I do is, from a lactating mother, I would pick up the gene which codes for human alpha lactalbumin. I will transfer this into a cow, because I'm drinking the cow milk on a daily basis, into the cow, and especially into their protein coding area. Okay, so let me look into it. This is my cow's gene. Okay, it's a huge structural gene in there. Okay, so this is the cow's structural gene, which will be coding for their own lactalbumin. This is not mine. Now what I do is, this is getting inserted. So let me insert that here. Now this is my alpha lactalbumin. Now if I take this milk, I will be having the enough or the enriched milk. Now this is my enriched milk. I keep drinking this, I'll definitely develop my muscles. I'll be more strengthened, you know. So the gym person would say that, oh wow, you have improvised. So yeah, so coming back to that point, let's look into it. So we only thought about introducing this into the cow. So which cow? We made that a cow with a name, and that cow was Rosie, and it was born in the year 1947. And then comes a very important question: how much per liter would you get this alpha lactalbumin? How much per liter you would get? And you'd say that it is 2.4 grams per liter. That is how much you would get from this milk. If you drink the enriched milk, you will get 2.4 grams per liter. So that's how you get it from the Rosie, and that's why it is very important also. So I hope the transgenic cow is very clear to you. So from there, we move on to the last topic of our class, and that is alpha-1 antitrypsin, which is famously known as AAT. The question is, where is this AAT being formed? This AAT comes from our liver. So our liver produces this enzyme AAT. This comes into the C part, that is our duodenum area. Let's look at the duodenum. So from here, AAT makes its way all to the duodenum. So inside the duodenum, like I said, there will be the secretions from the pancreas too. So let's pick up the pancreas. So this is my pancreas. Clear? And this pancreas will be giving in the enzyme called as elastase, and also have the enzyme called as trypsin. So these two are the enzymes that the pancreas are giving, and AAT is coming into the duodenum also. So once these two are there, if suppose you have consumed something which is in cartilage level, or you have consumed something which is in very long proteins, then trypsin and elastin will be activated. So once the food is digested and passed on, then AAT is released, so that AAT makes sure that elastase and trypsin is being blocked or inhibited. So what is the role of AAT? AAT inhibits trypsin and elastase enzyme. The point is that if AAT is deficient, then what will happen? Elastase enzyme will be increasing. Now, if this increases, the issue is it will move towards the lungs. So see, it's moving towards the lungs. So elastase will move towards the lungs, and inside the lungs we know that there is the alveoli, right? This alveoli is having the connective tissue layer also. So inside of this you will have the elastin fibers. Now during respiration we learned that these elastin fibers are the one that gives the contractile elasticity for alveoli. That's why it is able to contract and relax smoothly. Now, once this is lost, elastin fibers is lost because it gets digested. How? Because of the enzyme elastase. So if elastase is there, it will lose this contractile property or the elasticity, and loss of elasticity is what we call as emphysema. Oh my God, another disease. So once you lose this, then your alveoli will be comparatively distended like this. See that? The blotches in there. So we get the emphysema in there. Now the question is, how do I cure this? Because see, this is not because of smoking. Normally we say that emphysema is locked along with the nicotine coating, but this is not the case in here. This is just because a kid is born with an AAT deficiency. So we are having the elastin fibers getting digested off. So the only way is to replace this AAT which is lost in here. So what I do is I take up the AAT from a fresh liver cell. I take up the gene which codes in for AAT. So I take it from the liver. Okay. I got the gene. Now this gene will be transferred into the protein gene, okay, the lactalbumin. Gene. So this is a gene which I'm getting coded in here. This gene comes in here, okay, and this gene is of the goat, or you can put it as a sheep. It's okay. So the goat or the sheep is being with the alpha lactalbumin, and to this we are adding on this, which means that the coding will be into the milk. So this goat milk will be now rich with AAT, and such a goat which has been produced is called as Tracy, and that was in the year 1993. So by drinking this goat milk, we will be cleared or will be cured from emphysema. That is how you do it in the case with small children who are suffering from emphysema, because they did not smoke but they contract the disease. So thank you, and that was a very valuable lesson in there. I hope all the points are very clear. If you have any doubts, you feel free to contact me or try to connect with us in the future too. So thank you, and this is everything from Sapphire. Thank you.